In this lecture, I will walk through the statistical foundation behind eQTL mapping using single-cell RNA-seq data. We'll build up the ideas step by step, starting with what makes single-cell data special, then working through the statistical challenges it creates and how we solve them. Let's get started. So, what single-cell RNA-seq gives us? In bulk RNA-seq, we measure the average gene expression across all cells in a tissue sample. We get one number per gene per donor. Single-cell RNA-seq is different. It measures each cell individually, giving you thousands of measurements per donor per gene. And importantly, those cells are not all the same. They can differ in cell type, cell state, where they are in a cell cycle, how activated they are, and more. All of that variation is now visible with single-cell RNA-seq. Taking whole blood as an example, it contains many distinct immune cell types: B cells, T cells, monocytes, NK cells, and more, each with its own transcriptional program. Single-cell RNA-seq can separate all of them from a single sample. This means instead of asking, does this variant affect expression in blood, we can now ask, does this genetic variant affect expression specifically in monocytes or only in activated T cells? Questions that bulk data simply cannot answer. Here's a concrete illustration. A genetic variant shown as TT, GT, or GG genotypes, as we see that the G allele increases by 1 in each group, and might have a strong effect on expression, specifically in monocytes, but no effect at all in T cells and B cells. If we average across all cell types together, as bulk RNA-seq does, the monocyte signal gets diluted by the flat signals in all the other cells and disappears entirely. Single-cell resolution simply lets us see it. So, to summarize this first session, bulk eQTL mapping uses averaged expression and can miss cell type specific effects. Single-cell eQTL mapping uses individual cell measurements and can uncover effects hidden in the bulk average. The question now is, how do we statistically model this single-cell data? And the simple approach, and that is still widely used, is called pseudobulk. The pseudobulk approach is quite straightforward. For each donor and each cell type, we aggregate individual cells' read counts into a single summary, typically the sum or mean. This gives one value per donor per cell type, which we can then analyze with the standard linear model, as we discussed in a previous lecture for bulk eQTL mapping. It's simple, fast, and computationally efficient. But collapsing thousands of cells down to a single number comes at a cost, and those limitations matter. There are several limitations of pseudobulk. We're listing them here. First, it does not model the distribution of gene expression across cells. A single mean or sum may not accurately capture what's happening. For example, if half the cells express a gene highly and half express nothing, the average looks moderate, but the true picture is bimodal. Second, cell-level covariates are lost. If we want to account for where a cell is in a cell cycle or how activated it is, we need cell-level measurements. Pseudobulk has already thrown that away. Third, it does not account for cell number imbalance. A donor with 50 cells and a donor with 5,000 cells contribute the same single value, ignoring the very different reliability of those two summaries. These limitations motivate models that work directly on individual cells, which is what we build toward next. Single cell expression values are counts, non-negative integers, often with many zeros. The standard linear model assumes continuous, normally distributed outcomes, which is a poor fit for count data. The right model here is Poisson regression, in which the log of the expected count is modeled as a linear function of genotype and covariates. The genotype effect beta 1 is on a log scale, which gives it a natural interpretation. Each additional effect allele multiplies expected expression by e to the power beta 1. So if beta 1 is 0.7, that's approximately a twofold increase per allele. A fold change maps directly onto how biologists think about gene regulation. We also add a library size offset to correct for sequencing depth, because a cell with more total reads will show higher counts for every gene, and that's a technical artifact, not biology. Now, here's another structure problem we haven't addressed yet. In a single cell data, each donor contributes hundreds or thousands of cells, and all of those cells share the same genome, maybe similar environment, and batch effects. They are correlated. If we treat each cell as an independent observation and run Poisson regression naively, we're inflating our effective sample size, and the result is p-values will be far too small and will have a massive false positive rate. And the solution is a mixed model, which explicitly models the correlation structure within donors. As here is the Poisson mixed model. It solves the issue by adding a donor-specific random effect, B sub I, to the linear predictor, which is highlighted in the yellow box here. This term is unique to each donor and accounts for the shared variation among their cells. The fixed effects beta 1 for genotype still estimates the population level genetic signal we care about, and the random effect follows a normal distribution with mean zero and variance sigma squared times the covariance matrix big sigma. As we show in the matrix here, it can help capture the correlation of cells from the same donor. And in plain terms, the mixed model anchors the test to give correctly calibrated p-values while modeling gene expressions at the cellular resolution. So here we introduce eQTL, which brings together everything we've just covered into a single scalable framework to map eQTLs using single cell data. It is a toolkit that we recently developed and is still under continuous development. The challenges on the left map directly to the solutions on the right. Correlated cells are handled by random effects in the mixed model. Sparse count data is handled by the Poisson regression. Millions of cells and variants are made tractable through computational optimizations inherited from the Sage software developed for biobank scale GWAS. And in addition, there are two features that are added that can largely benefit the single cell eQTL mapping, and context-dependent effects can be tested via a genotype by context interaction term. And, and for the rare genetic variants, where the individual or single variant tests are underpowered, they are grouped together in a set-based test to improve power. So together, this makes eQTL an appropriate tool for single-cell eQTL mapping at scale. And here I'm showing the input and the model demonstration of eQTL used, and it can be used to detect three classes of genetic effects on gene expression using single-cell data, common eQTLs, and rare variant eQTLs, and context-dependent eQTLs. So for each gene and variant level p-values are combined to identify E genes, which are the genes that those expression is significantly regulated by genetic variations. So let's close this section with a concrete result. In the 1K1K cohort, where which profiled 1,000 donors across average for each donor, 1,000 immune cells at single cell resolution. CHQTL identified roughly 48% more E genes than pseudobulk approach. And this gain comes directly from the statistical machinery we've built up, modeling cells individually rather than aggregating, using Poisson likelihood appropriate for count data, and correctly accounting for cell level correlation through the random effect. That means more biology from the same data are obtained with better calibrated tests.